Cynocean nebulosus, more commonly known as spotted sea trout, are an important game fish in South Carolina. Approximately 90% of the adult population of spotted sea trout are infected with a myxosporidian parasite, Cudewa inornata. These parasitic cnidarians have been observed to cause cysts and reduce fishing and aquaculture productivity. Cudewa inornata have not been observed to cause any detrimental effects on sea trout. Instead, studies have found that, contrary to normal parasitism, sea trout infected by Cudewa actually swim faster in short bursts compared to uninfected fish. The purpose of this study is to evaluate the role of temperature in this relationship and to better understand the mechanism of infection. As climate change persists, understanding the relationship between temperature increase and parasite function, as well as giving a better definition of host-parasite interactions between these species, will be beneficial. This study set out to assess the effects of temperature on the metabolism of spotted sea trout and to examine the relationship of parasite load on metabolism of spotted sea trout acclimated to different temperatures. We hypothesized that metabolism and parasite load would be affected by changes in temperature. In order to assess these objectives, the following methods were used. Juvenile sea trout were transported from the Waddell Mariculture Center to the Marine Resources Research Institute in Charleston, where they were experimentally infected with Cudewa inornata via exposure to raw Charleston seawater. Sea trout were then transported to indoor and outdoor aquaria at the South Carolina Department of Natural Resources, as shown in the video. Following transport, sea trout were experimentally acclimated to two temperature variants of 12 degrees Celsius and 30 degrees Celsius. These temperatures were chosen to simulate high and low temperatures in Charleston Harbor. Fish were held for 2 weeks in an environmental room at the given temperatures. Our lab was in operation during the coronavirus pandemic. Unfortunately, we experienced some losses in our control group held at 12 degrees. This will later be noted in data processing. After 1 week of acclimation, a pre-swim blood draw was collected from the caudal vein of the fish, circled in purple. Hematocrit tubes and red blood cell and plasma samples were obtained. After 2 weeks of acclimation, spotted sea trout underwent exhaustive swim trials. Sea trout were put in a 100-liter volume tank and chased until exhaustion, which was characterized by the ability to tap the tail of the sea trout 3 consecutive times. Following swim trials, tissue collection occurred. Blood samples were drawn from the fish, and the brain, heart, white muscle samples, liver, and gills were dissected and collected for deep freezing in liquid nitrogen for future use and storage. Following blood collection, hematocrit samples were centrifuged to separate plasma from blood cells, then measured to determine red blood cell percentages. Additional blood samples were centrifuged at 5,300 G for 10 minutes at 4 degrees Celsius to separate blood and plasma. Plasma was collected and used for glucose assays and L-lactate assays, while red blood cells were deep frozen in liquid nitrogen for future analysis. Hematocrit testing yielded the following average percentages of red blood cells. 2 sample t-tests, assuming unequal variances, concluded that there was a significant difference between pre- and post-swim infected individuals, and post-swim individuals at 30 degrees and 12 degrees Celsius. This suggests that following swim trials, hematocrit percent is lower, and individuals at 12 degrees Celsius have less hematocrit than individuals at 30 degrees Celsius. Following hematocrit analysis, Eaton bioscience kits and a spectrophotometer were used to run L-lactate and glucose assays and determine enzyme concentrations. Concentrations of L-lactate in the 30-degree infected individuals and 12-degree infected individuals were found to be significantly different by a 2-sample t-test assuming unequal variances. This investigation gave us insight into different blood biochemistry parameters of spotted sea trout infected by Cudewa inornata in different temperature variants. We accepted our hypotheses that temperature and parasite load affect metabolism due to the results from significance testing. Further investigation into the role of L-lactate in swim performance in spotted sea trout and the health implications of decreased hematocrit and changing temperatures will help maintain spotted sea trout populations infected by this parasite. I would like to thank Dr. McElroy, Dr. George, and Dr. DeBuren for setting up the background research for this project. Additionally, I would like to thank the South Carolina Department of Natural Resources as well as the College of Charleston Grice Marine Lab. Last, I would like to thank the Beers Lab Group, Dr. Beers, Jessica Daly, Augustus Snyder, Jacob Cajor, and Riley Talmadge.